Hello students. Hello students. Today we'll study cell structure and function. Okay. So in lecture 1, first we are going to introduce with the cell. Okay. So this lecture only consists of the introduction to cell. First, what is the cell? So let me take the pointer first, then we will start. Fine. So cells are fundamental structural units of living organism and basic units of life. So before going to the definition, I can say you one thing. We have seen different flowers. We have seen different flowers, different plants, leaves, and different parts of the like different parts of plant, right? Like that, we have seen different parts of animals also. Okay, animals also we have seen. Now, all these parts, all these parts of the living organism that consist of cell, that consist of cell, and cell is the basic thing of the living organism, and this is the fundamental structure. That's why it has been told this is the fundamental structure units of living organisms or basic units of life. Without cell, we do not have any life. That's why it is the basic units of life. And cells are the fundamental structural units of living organism. Now, I can ask one thing. Who invented, ma'am? Or you can ask this question. Who invented, ma'am? Now, in 1665, an English scientist named Robert Hooke used a microscope to investigate the structure of thin slice of cork. He has taken a thin slice of cork. Okay. And here, after that, what he has done, that after that he has just tried to find out what it has. So he found that there are some in cork in woody tree. He just find out some part of this, that is the some dead plant cells of the cork he has observed, right? And Robert Hooke, who has just taken the cork under the microscope, he has seen dead plant cells of the cork, and the microscope used by Robert Hooke was primitive. And when better microscope has been invented, and after that magnified all these things, then scientists, they also investigated some pieces of the living plants also. So this, he has taken just a non-living cell, so non-living part. Okay, but living plants also, after the invention of different advanced microscope, that plant cells, the living plant cells, that has been investigated. Now the living plant cells that not empty like the cork cells. Here he has seen that this is like empty. Okay, but the living organism, if that has been taken, the living cells or living plant cells, and it has been seen that it is not like the empty like the cork cell, and they were found to contain a number of tiny structures called the organelles, okay, called the organelles. Okay, so that means here you can see that yes, there is an invention of the cell at that time. So the cells have been present in the living organism of the plant and animals since the origin of life. So in this way, it has been seen that there is a cell inside the cord. Inside the cord there is no cell, but inside the living organism or inside the slice of the living organism, you can see that there is some cells. Okay, so After that, this is the picture of the cell, and all these parts we will discuss: the nucleus, the endoplasmic reticulum, cytoplasm, cell membrane, DNA, lysosome, ribosome, mitochondria, Golgi apparatus. All these parts we will study here. Okay. Now in this part you can see that the pictures also given here. Let's start the definition. So what is the definition? The definition, that is nothing but the cell is defined as the smallest basic unit of life, that is responsible for all life's process. So that is responsible for all life process. That means this is the basic structural unit, or I can say this is fundamental unit of any organism, living organism. Okay, and this is the cell. Now the cell are the structural, functional and biological units of all living beings. A cell can replicate itself independently. So this is building blocks of life. And the shape, if I tell the shape and size of cell, it is different for size of cells. These are different for different types of organisms. So suppose you have one plant leaf. The cell look like this. This is a plant leaf. So you can see that the cell, if you see the cell here, so that is different. Whenever you are just taking one flower, you can see that will be another different thing. Whenever you are just saying one person here, at that time the cell structure that will be different, and the function, this is the functional unit also. The functions of the cell are different for different organs. The cell for the leaf, it has different types of functions. The cell for the flower, it has different. Also you can see that in our human being, the cell's functions are most complex thing, and in every organs of our living human being we have different functions. Okay, so cells are structural, functional and biological units of all living beings. Biological units of all living beings. Okay. Now, what the cell has? First thing, the cell, here you can see that for any cell, there is cytoplasm. Okay. So each cell that contains a fluid, that is called cytoplasm, and which is enclosed by a membrane. Now it is enclosed by a membrane. Okay. So that is called cell membrane. And this is the cytoplasm. Okay. Now this is the cytoplasm here. So what we have studied, that the cell that is filled up of this, what? This is a fluid that is called the cytoplasm, and it is enclosed by what? Enclosed by cell membrane. Now the cytoplasm has several biomolecules like proteins, nucleic acids, lipids. Now one thing is very sure, that cell is the basic structural and functional unit. Now to do any function, whatever we require to do any function, we require the cell should have some biomolecules, and it consists of proteins, nucleic acid and lipids. What it consists of? Proteins, nucleic acid and lipids. Okay. So cellular structures called cell organelles and are suspended in the cytoplasm. So here you can see that the cellular structure, that called cell organelles, and that is suspended in the cytoplasm. So first what we have studied, yes, there is a cytoplasm. What is cytoplasm? The cell consists of, that contains a fluid, that is called cytoplasm. Now this cytoplasm, this is enclosed by cell membrane. Okay, and this cytoplasm, it has what? It has several biomolecules, and it like protein, nucleic acid, and lipid. Protein, nucleic acid, and lipid. So whenever we are saying that any organ is doing its job or its function, the cells actually, they are doing the job, they are doing the function. So it also the cytoplasm that consists of biomolecules, several biomolecules, and these biomolecules like proteins, nucleic acids, and lipids. Okay, and the cellular structure that called the cell organelles and suspended in the cytoplasm. So what is cell? A cell is a structural and fundamental unit of life. We have understood, we just understood this thing because the structural means and the fundamental means that we studied already. The study of the cell from its basic structure to the function of every cell organelle is called cell biology. So a big part of this we are studying now. That means the subject which we are using to study the basic structures and also the functions of every cell, functions of every organelle, that every part of the cell, that is called cell biology. And Robert Hooke was the first biologist who discovered the cell. Already I have told you the main thing, that first he has taken one cork and just there is a microscope, and under this microscope he has just placed the cork, and he has seen at that time just seen one empty area. But whenever the microscope, this invention, now that means better microscope we have at that time, again the experiment has been done with the help of the living organism. Okay, at that time it has been seen that there is no empty, there is some small some pores are there, or cells are, some structures are there. So that is the cell. So Robert Hooke, he was the first biologist. He was the first biologist. Please note down this. Robert Hooke. Robert Hooke, his name. Please note down this. He was the first biologist who discovered cells. Okay. So cell is the fundamental or structural unit of life. Cells are the basic structure, and from the structure to function and types, all these things we can study, and that subject is called cell biology. And Robert Hooke was the first biologist who discovered the cells. Now, all organisms are made up of cells. It may be for human being, it may be for dog, it may be for plant, it may be for any microorganism also. They have also the cells. Now they may be made up of single cell, that is called unicellular, and another cell, one is the unicellular and another is the multicellular. Okay, so unicellular means only single cell will be there, and many cells if we have, then it is the multicellular. And mycoplasmas are the smallest known cells. So which one is the smallest known cell? Mycoplasmas. These are the smallest known cells. So cells are the building blocks of all living beings. We know that thing, that to provide any structure of body, convert the nutrients taken from the food into energy, the cells are doing a lot of functions. So from the nutrients, whatever we are just taking as a food, from that, the energy conversion, that also we require the cell. So there are a lot of functions of the cell that we will study. Now cells are complex and the components perform various functions in organism, and the complexity of the cell that depends on the different types of animals or plants, right? In human being, the cell organism, it has most complex structure, and they are different shapes and size, pretty much like bricks of the building. And our body is made up of several cells. Our body is made up of several cells, and it is different shapes, and it has different sizes also. Different sizes also. Now, cells, they are lowest level of organization in every life form, and this, from the organism to organism, the count of cells may vary. That means if I am calling unicellular, that means this is related to very small or tiny organism. But if it is your multicellular, at that time we are saying it is depends on the different human being or different animals also or plants also maybe. So humans have more number of cells compared to the bacteria. So that means from microorganism they have very small amount of cells, but human being, this is the most complex. Already I have told the human body, this is the most complex thing. So here the number of cells are huge. A huge number of cells are there because huge number of functions we have to do. Now, cells comprises of several cell organelles that perform the specialized functions to carry out life processes like food to energy and different activities also. They have to do. So if the any organ, it has to do some different activity, that means it's the cells actually it is performing the main function. So every organelle, it has a specific structure, and hereditary material of the organisms is also present in the cell. That is the DNA. Okay. So it is coming from hereditary material. This is the hereditary material. Now, who discovered cells? Already I have told, Robert Hooke discovered cell in 1665. And Robert Hooke observed a piece of bottle cork under a compound microscope and noticed that minuscule structures that reminded him of small rooms, and consequently he named these rooms as the cells. And his compound microscope had limited magnification, so he could not see the any details structure of this. Now, owing to this limitation, who concluded that these were non-living entities. The cork was non-living entities. So he has noticed some minuscule structures. Okay, and it is like small room. So he has given the name, that is the cell. Now later Anton van Leeuwenhoek that observed the cells under another compound microscope, and it is a little bit higher magnification, and this time the cells exhibit some form of movement. So as a result, Leeuwenhoek concluded that microscopic entities were alive. That means cells are alive. So after a host of their observation, these entities were named as animalcules. Okay. So these are alive. So in 1883, Robert Brown, a Scottish botanist, provided the very first insight of cell structure. So who has seen the Robert Brown, the Scottish botanist? Okay, so Scottish botanist, he has seen some very first insight in the cell structure, because before that the whatever the microscope, they are not high definition microscope and they are not high magnification is possible, so they cannot see the proper structure of the cell. But now Robert Brown, the Scottish botanist, he has seen, yes, their structure of the cell also he has observed, and he was able to describe the nucleus present in the cell orchids. So first thing has been introduced in the book, that is the nucleus. He was able to describe the nucleus present in the cells of orchids. He has taken the orchids. Now the characteristics of the cells, right? So already we have studied Robert Hooke. He has first taken the cord. Now he was just unable to see the whole structures, but he has seen some small rooms and he has given the name, that is the cell. But after that, it has been done that due to the limitation of the magnification of the microscope, what happened after that, the total structure cannot be analyzed. So in the 1883, Robert Brown, he has just seen that yes, there is there is some life, there is some live structures are there, and nucleus also that has been presented here. Okay. Now the characteristics of the cells. Here, cells provide the structure and support to the body of organism. So, structure and support, right? For every organism, it provides structure and support. Cell interior is organized into different individual organelles surrounded by separate membrane. In this cell, whatever the cell organelles you have the cell membranes there, right? So now the nucleus holds the genetic information necessary for reproduction and cell growth. So the nucleus it holds the genetic information necessary for what? For reproduction and growth and growth. Okay, and growth. And every cell has one nucleus. Every cell has one nucleus and membrane bound organelles in the cytoplasm. And membrane bound organelles in the cytoplasm. Now, next is the mitochondria. This mitochondria is a double membrane bound organelle that is responsible for energy transaction, and it is very vital thing for the survival of the cell, right? So in the cell, first we have cytoplasm that is made up of several biomolecules, and it consists of lipid, protein, and also nucleic acid. And the cytoplasm, it is just surrounded by what? That is the cell membrane. And in the cell also we have the nucleus that holds the genetic information necessary for what? Necessary for reproduction and growth. And every cell, it has nucleus and membrane bound organelles in the cytoplasm. Next is the mitochondria, which is the very important thing of the cell, and which is very vital for the survival of the cell. And it is a double membrane bound organelle. It is double membrane bound organelle, and it is responsible for the energy transaction, right? So what we have studied till now? Cell, discovery of the cell, and then the cytoplasm, then the cell membrane, nucleus, then we have different membrane bound organelles and mitochondria, right? Next, lysosomes. This digests the unwanted material in the cell. This digests the unwanted materials in the cell. And endoplasmic reticulum, that plays a significant role. Endoplasmic reticulum, it plays a significant role in the internal organization of the cell by synthesizing selective molecules and processing, directing, sorting them to their appropriate locations. So lysosomes, they are doing the digestion of unwanted materials in the cell. Okay, digest the unwanted materials in the cell. And endoplasmic reticulum, endoplasmic endoplasmic reticulum. Endoplasmic reticulum, what it will do, it will just take the part in the internal organization of the cell by synthesizing selective molecules and processing. It will do the processing, directing, directing and sorting, and sorting to them to their appropriate locations. So several molecules are there. So for processing, directing, sorting, all these molecules, it will do that work. Okay. So world of cells, prokaryotic cell and eukaryotic cells. There are two different types of cells here. So according to the number of cell, we can say that the cells may be of two types, unicellular and multicellular. But according to nucleus, we can say there are two types. One is prokaryotic, another is eukaryotic. Eukaryotic cell. Now, prokaryotic cell, it has generally small 1 to 10 micrometer, where 1 micrometer that equals to 1 micrometer that equals to 10 to the power minus 6 meter. 1 micrometer that equals to 10 to the power minus 6 meter. And the nucleus is not well defined and known as nucleoid. And single and circular chromosome are there. Membrane bound organelles absent. Example is the bacterial cell. So membrane bound organelles are absent here. Okay. So this is prokaryotic. We will study elaborately that prokaryotic. Now the prokaryotic cells, so it has no nucleus. And instead of nucleus, the prokaryotic such as bacteria have a region within the cell where genetic material is freely suspended. And this region, that is called nucleoid. That is called nucleoid. Okay. So there is no nucleus in the prokaryotic cell, but there is a region within the cell which carries the genetic material and it is freely suspended. That is called nucleoid. That is called nucleoid. Now, this prokaryotic cell, these are single cell microorganism. That means unicellular, like archaea, bacteria and cyanobacteria. The cell sizes are 0.1 to 0.5 micrometer in diameter, where 1 micrometer equals to 10 to the power minus 6 meter. And the hereditary material can either be DNA or RNA. Okay, hereditary material can be DNA or RNA. And the prokaryotes, these are generally reproduced by binary diffusion, by binary fission, and a form of asexual reproduction. This is the asexual reproduction. We know, we have studied earlier, this is asexual reproduction. Asexual reproduction. And they are also known to use the conjugation, which is often seen as the prokaryotic equivalent to sexual reproduction. However, it is not the sexual reproduction. So the reproduction will be done by the binary fission and it is the asexual reproduction method. Now, prokaryotic cell definition, if we want to define, then prokaryotic cells, these are the cells where do not have a true nucleus and membrane bound organelles. So two things it does not have. One is the nucleus. Instead of nucleus, what it has? Nucleus. Okay. And it does not have any membrane bound organelles. Okay. Now, what is prokaryotic cell? So, till now we have studied prokaryotic cell. It is the single-celled microorganism and known to be earliest on the earth, and prokaryotic include the Bacteria, bacteria and archaea, bacteria and archaea. And the photosynthetic prokaryotes include the cyanobacteria that perform the photosynthesis. Now, prokaryotic cell consists of single membrane, therefore reactions occur within the cytoplasm. Within the cytoplasm, whatever the reactions according to the functions, that reactions will be performed in the cytoplasm. And they can be free living or parasites also. They can be free living and or we can say that parasites also. Now the characteristics of the prokaryotic cell. Prokaryotic cells have different characteristics, like they lack a nuclear membrane and mitochondria, Golgi bodies, chloroplast, lysosomes, nothing is there. Mitochondria, Golgi bodies, chloroplast, lysosomes, these are absent. And the genetic material is present on a single chromosome. The genetic material that present on a single chromosome, the histone protein, important constituent of eukaryotic chromosomes, are lacking in them. There is no histone protein. There is no histone protein. No histone protein that is present in the eukaryotic cell. And the cell wall is made up of carbohydrates and amino acid carbohydrates and amino acids. So the genetic material in the prokaryotic cell that is present on a single chromosome, the histone proteins, these are absent, which are present in the eukaryotic cell, and the cell wall that is made up of carbohydrates and amino acids. Fine. Next, the plasma membrane that acts as the mitochondrial membrane that carrying the respiratory enzymes. So the plasma membrane, this acts as the mitochondrial membrane, and they divide asexually by binary fission. Okay, and the sexual mode of reproduction involves conjugation. So prokaryotic cell structure, the prokaryotic cell does not have a nuclear membrane. We know that thing. There is no lysosome, and it may be spherical, may be rod shaped, may be spiral. A prokaryotic cell structure, first is the capsule. What is capsule? It is the outer protective covering that found in the bacterial cell, and we are calling this is the capsule. And it helps to protect the cell from moisture retention, protects the cell from engulfed, and helps the attachment of cell to nutrients and surfaces. So this capsule is very important part. Now the cell wall. The cell wall, that is the outermost wall layer of the cell, which gives the shape of the cell. It gives the shape of the cell. Then the cytoplasm. Cytoplasm, already we know that it is mainly composed of enzymes, salts, and cell organelles, and this gel-like component is there. Fluid is there, and it has different biomolecules, and it has enzymes, salts, like that. That means which is required for the, what are the nutrition required to be alive. Now the cell membrane, this layer surrounds the cytoplasm and regulates the entry and exit of substances in the cells. So this cell membrane, this is the main door. We can say it is the door, and this layer surrounds the cytoplasm and it regulates what? The exit and entry of the substance in the cell. So I can say that it is the door of the cell. Now the pili. These are hair-like outgrowths that attach to the surface of the bacterial cell. In the bacterial cell, we have seen that some hair-like, this type of hair-like outgrowths are there that attach to the surface to the other bacterial cells, that is the pili. And flagella, these are long structures in the form of whip. These are long structures in the form of web. That is the flagella. That is the flagella. That is the flagella. So here you can see that the flagella, these are the long structures in the form of a web, and here that help in the locomotion of the cell. So here you can see that suppose this is, and there is a flagella, and this helps to, this is the flagella, and it helps to move the, it helps to move that particular microorganism. Then the ribosome, then the ribosome. Okay, so ribosomes here, they are involved in protein synthesis, and ribosome, here you can see, if I just want to draw this thing, so this is a cell, and the ribosomes, there are small tiny, these are the ribosomes, and this is the nucleus, this one is the nucleus. Okay, so if these are the ribosomes, okay, and these are the ribosomes, these are the ribosomes. So these ribosomes, they involved in protein synthesis, involved in the protein synthesis. Next is the plasmids. Next is the plasmids. So the plasmids, plasmids here, the plasmids are non-chromosomal DNA structure, and these are not involved in reproduction. These are not involved in reproduction. So here if I draw one bacterial here, so you can see that, you can see these are plasmids. Okay, and this is the bacterial DNA. These are the bacterial DNA. Okay, so this is the plasmids. So plasmids are non-chromosomal DNA structure, and these are not involved in reproduction. These are not involved in reproduction. Now the nucleoid region, nucleoid region, nucleoid region. So here, this region in the cytoplasm where the genetic material is present. Nuclear region, here you can see that the genetic material will be present, and prokaryotic cell lacks certain organelles like mitochondria, endoplasmic reticulum, and Golgi bodies. So which are absent? That is nucleus and mitochondria. Sorry. Mitochondria and endoplasmic endoplasmic reticulum, endoplasmic reticulum, and also Golgi bodies. These are absent. Okay, so there is no nucleus, there is some nuclear region where the cytoplasm and it has some genetic material present, and mitochondria is absent, endoplasmic reticulum is absent, Golgi body is absent. Now, prokaryotic cell diagram, here we can see that the absence of true nucleus and the presence of flagellum, it differentiates it from the eukaryotic cell. Now, here you can see that diagram of the prokaryotic cells. So, till now we have studied one thing. This is the capsule. This is the capsule that gives the protection from moisture and other hazards also, right? And cell wall, then you have the cell wall. This cell wall, it binds the cytoplasm, and this is the plasma membrane. This plasma membrane, inside this you have some gel type liquid, that is the cytoplasm, gel type fluid. Okay, gel type, sorry, just a minute. Okay, gel type fluid here. So this is the gel type fluid and this is cytoplasm, and here you can see that ribosomes, these are ribosomes, these are ribosomes, and here DNA nuclear. Here you can see that DNA nuclear, but there is no nucleus. There is no nucleus. And here you can see that bacterium flagellum, this is used for locomotion. This is used for locomotion, right? So this is the plasma membrane. This is the cell wall, capsule, cytoplasm, ribosomes, mesosome, and DNA. Mesosome and DNA. And DNA. Okay. So these are the main structure of the prokaryotic cell, main structure of the prokaryotic cell. So here you can see that the mesosome also, this mesosome, here I am writing mesosome, I have not told the mesosome. This is a specialized membranous structure. This is a specialized membranous structure which is formed by an extension of plasma membrane into the cell. Okay. So these mesosomes, you can see that the plasma membrane here, and here this is the plasma membrane. So this is a specialized membranous structure which is formed by an extension of plasma membrane into the cell. Here you can see this thing. Okay. So this is the mesosome. Now the components of the prokaryotic cell, just now we have seen the plasma membrane. It is an outer protective covering of phospholipid molecules, and it separates the cell from surrounding environment. It separates the cell from surrounding environment. Next is the cytoplasm. It is a jelly-like substance present inside the cell. All the cell organelles are suspended in. And next is the DNA. It is the genetic material of the cell. All the prokaryotic possesses a circular DNA. Like you have seen in the cell just now that there is some DNA like that. There's circular DNA. And it directs with what proteins the cell creates. And it also regulates the actions of the cell. Now the ribosome, protein synthesis occurs here. Some prokaryotic cells possesses the cilia and flagella, which helps for moving here and there, that is for locomotion. Now reproduction in prokaryotic cell, it may be reproduced in two ways. One is the asexually, that is the binary fission, and another is the sexually by conjunction, conjugation, okay, conjugation. So asexually binary fission and sexually by conjugation. Now binary fission, the DNA of the organism replicates and new copies attached to the cell membrane. It is very simple thing. So it is asexual reproduction. The DNA of the organism, it replicates and the new copies will be attached to the previous cell membrane. So here the cell walls, you can see that starts increasing in size and starts moving inside. So binary fission, here you can see that always what happens here. Here you can see that first in the binary fission, you have some cell here, then it will be like this. After that, this will be like this. Then what happens? Two cells are attached here. Then you can see they will be separated. They will be separated. Okay, so this is the bacterial binary fission. So DNA of an organism replicates and the new copy is attached to the cell membrane. The cell wall starts increasing in size and starts moving inwards, and the cell here you can see that the cell membrane it starts to moving inwards here, and the cell wall is then formed between each DNA, dividing the cell into two daughter cells, and here these are the daughter daughter cell. So this is also another daughter cell. Okay. Now recombination. In this process, genes from one bacteria are transferred to the genome or to the other bacteria, and it takes place three ways: conjugation, transformation, and transduction. Conjugation, transformation, and transduction. Conjugation, this is the process in which genes are transferred between two bacteria through a protein tube structure called phylus. So conjugation, here you can see that for the conjugation, the process in which genes here are transferred between two bacteria through a protein tube structure, So this conjugation is a form of, the conjugation is a form of sexual reproduction in bacteria, and two individual cells are united by a tube. Here, suppose this is a cell and this is another cell. This is another cell you have, and here you can see that this will be, at last this will be conjugated. This will be conjugated, and here okay, so here this will be transferasome and this is the relaxosome. So after that you can see that the new donor will be produced. Okay, so first we have the chromosome of bacterium and the pilus, then donor and recipient we have, and they are attaching to each other. After that new donor will be come out. Okay, so this is the conjugation. Now the transformation, it is the mode of sexual reproduction in which the DNA from the surrounding is taken by the bacterial cell and incorporated in its DNA. And transconduction, this is the process in which genetic material is transferred into bacterial cell with the help of viruses. And bacteriophages are the virus that initiates the process. Now examples of the prokaryotic cell, first is the bacteria cell. bacterial cell. And some unicellular organisms also we can found in the earth from the soil to human body, these prokaryotic cells. And they have different shapes and structures. The cell wall that is composed of peptidoglycan that provides structure to the cell wall. And bacteria have some unique structures such as pili, flagella and capsule. Pili, flagella and capsule. Pili, flagella and capsule. Okay. Pili, flagella and capsule. Okay. This much for today. Thank you.